The question is, how can we use liquid biopsies to fit into cancer research? And I think we're all pretty excited about that. Frankly, the advantages of a so-called liquid biopsy is that it's a blood test, as opposed to tissue, which requires a biopsy. So biopsies are more expensive. They are usually have to be invasive because although there are a few people who have lumps and bumps that are easily accessible, most of them are obviously in the liver or the lung or whatever. And so while that's not impossible to do, it becomes logistically and also maybe dangerously difficult to do serial biopsies. The other advantage of a so-called liquid biopsy is that it may give you a snapshot of the entire patient's cancer load as opposed to a biopsy, which is just the site you stick the needle in. In fact, I think these are complementary. We haven't proven it yet, but I believe what we'll see are things we can pick up with a real biopsy. And one advantage, of course, is you get many more cells. You get 1 billion cells in a biopsy, so you don't have to sequence as deeply. The cancer cells are right there. They're not rare events. So that's an advantage there, whereas the liquid biopsy, either the cancer cells or the cell-free DNA are really rare events. So you've got to sequence pretty deeply to get to them, so they're less sensitive than a true biopsy. So I think these are going to complement one another. Now the issue is, how do we use them in research? There are a number of ways. One would be because you believe that patients who don't have whatever the marker is are so unlikely to benefit or need it that you shouldn't put them on trials. We've been doing that for years. For example, in breast cancer, we don't put estrogen receptor negative patients onto trials of endocrine therapy. Good example. The second would be that you actually use it to identify a marker that you think the drug will work. So that's called predicting it will work. And so that would be eligibility. Again, I'll use the same example. We put ER-positive patients onto clinical trials of new endocrine therapy because we believe it's the target for the endocrine therapy. In fact, we don't believe, we know that's true. So those are two different ways you could use it just to make your trials more efficient. If you do trials in patients who either don't need it or are unlikely to benefit from it, you'll dilute out the true effect and get a false negative. So you don't want to do that. Also, you spend a lot of money in patients where it won't work. And you expose those patients to drugs that won't help them, and that's not the way we want to do things. The second thing would be to use a circulating biomarker as an endpoint itself, as a surrogate. So let's face it, the most objective and meaningful endpoint is survival. Survival is very easy to measure, but it takes a long time. And so if you want to do a relatively quick study, you don't want to wait and see if patients are alive or dead. The next surrogate in from that is progression-free survival. Progression-free survival is actually difficult to measure in terms of looking at radiographs and that sort of thing. So it'd be nice if you had a circulating marker that either went up or went down and reliably predicted what was going to happen to the patient for the other two endpoints down the road. That could be easily collected and measured. Again, I've gone through why I think liquid biopsies are good. And this is also not unprecedented. For example, in prostate cancer, we've used PSA as a measure of response or not. In ovarian cancer, we've used CA-125, and so on and so forth. And now increasingly, we've been involved with looking at circulating tumor cells. It's pretty clear that if you have if you're given chemotherapy and we look at circulating tumor cells one cycle in, if you haven't reduced those down below a certain level, you're almost certainly not going to benefit from that chemotherapy, and you need something else, as an example. And then the third thing is whether or not you could actually use liquid biopsies sequentially to begin to identify the biology of why this drug isn't working. So you might begin to see emergence of mutations or phenotypes, for that matter. Likewise, you might see that in patients who do well, they don't have emergence of new markers, and that might give us a way to to explore whether or not other therapies could be added in or substituted for the drugs you're using. That's in the future, but I think all of us believe it's going to happen. The final way that I believe they could be helpful is as a pharmacodynamic monitor in early diseases, so in early trials. So for example, if I have a down-regulator of estrogen receptor and I have circulating tumor cells that make estrogen receptor, perhaps maybe within 2 weeks or a month, I could begin to see whether or not whether I'm at the right dose for that drug, if the estrogen receptor is decreasing or disappearing in the circulating tumor cells that are left behind. That hasn't been proven yet, but I think it's one of the ways many of us believe it's possible. People have tried to do this in white blood cells with other kinds of drugs that downregulate things. Or the flip side of that would be whether or not you believe the drug causes some change in the pattern of genetic expression, and you could measure that going down. There are a number of people who are trying to do these sorts of things. All told, I think we can use these things in a very smart way to make our trials more efficient, and that way we can get to the answer that our patients want, which is, are these going to make them live longer or feel better faster than the way we do things now? You know, it's interesting to me, one of my friends, Mark Lippman, often says if you wait for 10 years, you can do what somebody else has done and call it something new and pretend like you made it up. That doctors have been trying to deliver personalized medicine with precision since the time of Hippocrates. This is not new. What's new is we have better toys and better tools to deliver our personalized care with more precision. So unless you're a public health doctor, where, you know, like for example, sewers aren't personalized care, right? But they probably have had and vaccines have had the most impact on health in more than any drug we probably have, if you think about it. But as an individual doctor, we are trying to personalize our care with as much precision as possible. And I think that this is one step forward to becoming even more personal with more precision. Ultimately, the issue is whether we will get down to only treating one patient with one drug or one set of drugs, or still have groups. I think we'll still have groups of patients. We're not going to be able to get down to the individual, I don't think that much. This gets into heterogeneity, which has been recognized for 150 years as a big issue in cancer, and for the last 50 or more years as a big issue in resistance to therapy. And there are two issues. One is inter-patient heterogeneity, so that one patient is in breast cancer ER positive, the other patient's ER negative. We call it breast cancer, and yet those are two different diseases, and I can go on and on and on. The other, for an individual patient, is intra-patient heterogeneity. And again, this is not a new recognition, but we have better tools to begin to recognize. In fact, we've ignored this in many ways. We've known about it. It's been written about for decades, but we've just gone ahead and treated people. But I think we can do better. So for example, if we get back to liquid biopsies, we and others have seen that in a patient who is called, I hate it when people do this, but it's called HER2 or ER estrogen receptor positive, that individual circulating tumor cells, some will be estrogen receptor positive, some will be in between, some will be completely negative, same patient. Well, we've known that in tissue, but we can begin to see that evolve going, for example, we've seen patients who 99% of their primary cancer is ER positive and really hot ER positive, whereas by the time they have metastasis, their circulating tumor cells, maybe 10% are very hot and the other 90% are not, and so on and so forth. We don't know actually that predicts what's going to happen to those patients. That's what we're starting to do now, is saying, okay, now that we can do this, let's do trials and see whether or not it actually predicts resistance to therapy. Or better yet, begin to see if you treat those patients with endocrine therapy, do they begin to evolve even more into ER-negative cancers? Or for example, the estrogen receptor gene, ESR1, begins to develop mutations that make it resistant to one kind of therapy and not another. I think I'm using breast cancer as an example because that's what I do, but we're going to be doing this with every disease now. I think we're going to be able to start sorting out the biology and the heterogeneity of that biology disease by disease and case by case. And I think that's pretty exciting because I think it'll guide us then into using our so-called precision therapy with more precision, which is what we're trying to do.